Hi, my name is Nicole Barbour, and I'm originally from California, and I study movement ecology at the University of Maryland Center for Environmental Science. I actually met Dr. Helen Bailey here at the Chesapeake Biological Lab, and she had availability for me to study sea turtle migration movements. My research is really important because sea turtles are endangered, and specifically leatherback turtles are endangered, and these turtles are impacted by fisheries especially, which the interaction is happening in areas that we can't really control. They're happening in international waters, really far away from land. So the majority of conservation efforts have focused on protecting beaches and the leatherback nests that are on these beaches. But in order to actually conserve and protect these species in their primary habitat, which is at sea, we have to be able to understand their movements and be able to predict them in time and space. So CBL has given me a chance to do both fieldwork and modeling together, and to be able to also collect my fieldwork remotely. So my fieldwork is not in the Chesapeake Bay, it's actually in Central and South America. So I'm able to go out and collect my data seasonally, bring it back, and then analyze it and stay here. And additionally, I actually have 2 advisors, so I'm split between coming down to Solomons, where the Chesapeake Biological Lab is located, and being up in College Park. And so I get to take my classes in both areas through an interactive video network, and that's allowed me to make partnerships and relationships with people that I wouldn't have normally had access to. Our big question in this study was how oceanographic conditions are affecting the ability for these leatherback hatchlings to get to their offshore foraging habitat. And this is important because leatherback hatchlings, when they hatch, have a limited energy storage. Last summer when I was down in Pequara, Costa Rica, my project was focused on tracking hatchling, which are baby leatherback turtles. These tags are basically very small little acoustic tags. They have a very small weight, and so they don't actually affect the swimming ability of the hatchlings. And what they do is they actually produce a sound that's kind of like a ping, and we pick that up using a receiver, an acoustic receiver, which is deployed off of the side of the boat. And then we'd put them in the water, and then we would track them for about an hour and a half at a time from this boat from a very safe distance. So we performed this research in partnership with Upwell. In addition to that, this research would not have been possible without our other partners, including the Ecology Project International and the Nueva School. We are also super thankful for a lot of the volunteers that assisted us in this research, including Max Gotts, who contributed his photography and assistance in the field. Understanding hatchling survival is very important in understanding the survival of the population as a whole and being able to protect them moving forward.